Dear students, today we are going to study life cycle of Marchantia, which is a common bryophyte. The first point is occurrence. This Marchantia is one of the very important and common liverwort. Practically speaking, it is a cosmopolitan in distribution, and this Marchantia is widely distributed and commonly found in shady places, on moist soils, rocks, and other moist surfaces. In world, there are about 65 different species of Marchantia which are growing. Out of these 65 species of Marchantia, 4 species of Marchantia are commonly growing in India. These 4 species are Marchantia palmata, Marchantia nepalensis, Marchantia simlana, and Marchantia polymorpha. Out of these species of Marchantia, Marchantia polymorpha is a very common Indian species growing throughout the Indian continent. Then, structure of gametophytic thallus, or the external morphology of gametophyte of Marchantia. The gametophytic plant body of Marchantia is a thalloid, that is, it is undifferentiated into root, stem, and leaf, and this thallus is a dorsiventrally flattened. If you observe this thallus, it is a dorsiventrally flattened and prostrately growing with repeatedly dichotomous branching. If you observe, this is a first dichotomy, then the dichotomy goes on as one goes towards the tip of this thallus. It grows in the form of rosette. If you observe, these thalli, they are growing in a close vicinity and all they are arising from one point. So it results in the formation of a rosette structure or such a star-shaped structure. And if you observe, the apical end of this thallus, each such thallus is having a prominent notch, either at dichotomy or even at the tip of a lateral branch. Now, as I have told you that this thallus is a dorsiventrally flattened, that means it has a distinct dorsal and distinct ventral surface. On dorsal surface, you can have a prominent dark green midrib, and this midrib is present at the center of each lobe, and towards the apical region, this midrib again shows a dichotomy, and if you observe the upper surface or dorsal surface, along with the midrib, you can have a large number of asexual bodies which are called as a Zimmer cups, which are the asexual reproductive structure. Then the ventral surface, if you observe here, it has a number of rhizoids and scales. The position of rhizoids on the ventral surface is along the midrib region, whereas scales, they are present at the margins of the thallus. So this is a typical position. These rhizoids are root-like structures. That means they perform function of root. You know that root performs function of absorption, and it fix the plant body into the soil firmly, that is called as anchorage. So these rhizoids, they are absorbing structures, and the another function is that they fix this thallus on the substratum on which it is growing. These rhizoids are always unicellular. If you observe the enlarged view of each rhizoid, these are unicellular structure, that means there is no crossover. But based on the wall of rhizoid, there are two different types of rhizoids. One that is a smooth rhizoids or smooth walled rhizoids, and these are thin walled. Whereas in some rhizoids, the outer wall is a smooth, but inner wall shows presence of large number of infoldings, and these infoldings are called as trabeculae, and therefore these rhizoids are called as tuberculate rhizoids. In addition to rhizoids, another important structure that is present on the ventral surface of thallus of Marchantia is a scales. So these are very thin multicellular plate-like and violet colored structures present along the margins of this thallus on ventral surface. And the most important function of scale is the protection to the young growing tip of this thallus, and it is also assumed that it also helps in a transport of water or conduction of water. Thus, protection of the thallus from the mechanical injuries or other injuries, or protection of apical growing tip of thallus, and second one, that is the transport or conduction of water. So this is about the external morphology of gametophytic thallus, which is a dorsventrally flattened and dichotomously branched, repeatedly dichotomously branched, forming a rosette structure. The next important point is the internal structure of this thallus of Marchantia. If you observe this diagram, internally the thallus of Marchantia is clearly differentiated into two distinct regions. One that is present on the dorsal surface is a photosynthetic region, or which is also called as assimilatory zone or synthetic zone. Whereas below this photosynthetic upper zone, the second zone is a storage zone. That storage zone is present towards the lower or ventral surface of the thallus of Marchantia. So this is a very common and distinct organization of the thallus internally. If you observe the details of internal structure in a transverse section, here this thallus on a dorsal as well as ventral surface remains covered by means of a single layered epidermis. Epidermis, this is the upper epidermis, this is a lower epidermis. Upper epidermis is present on the dorsal surface and lower epidermis is present on the ventral surface. If you observe the upper epidermis, it shows the presence of such a air pores at a regular distance on the upper epidermis. So this air pore is directly open into a cavity that is called as air chamber. So large number of air chambers, they are present on the dorsal surface just below the upper epidermis. If you observe this air chamber, this is one air chamber, this is another, this is a third one. They are separated from the neighboring by means of a partition wall, that is nothing but a green parenchymatous, that is chlorenchymatous thin walled cells, which are called as a partition wall. Now, if you observe the details of this air chamber, you can have a large number of unbranched or branched photosynthetic filaments rising from the base of this photosynthetic zone, radiates in a erect fashion, and all these are commonly called as a photosynthetic filaments. Why they are called as photosynthetic filaments? Because each cell of this filament is a green and containing chloroplast and performs function of photosynthesis, and therefore these are called as a photosynthetic filaments or photosynthetic lamellae. Now, thus this entire upper zone performs the function of photosynthesis. Epidermal cells also contain chloroplast, that means they are green in color. The partition wall cells, they are also having chloroplast, and at the same time these photosynthetic filaments, they are also green in color, and thus they perform function of photosynthesis. So this entire upper zone or region is having a thin walled chlorine chimatous cells which performs the function of photosynthesis, and therefore it is called as a photosynthetic zone, or it is called as a photosynthetic layer. The main function of epidermis is a protection. Then this air pore forms a communication of this air chamber with the outer environment, that means it helps in a gaseous exchange and so on, and all these green colored cells that are present in this region are photosynthetic in activity. Then just below this photosynthetic zone is a storage zone. The cells of this storage zone are comparatively compactly placed, thin walled, and no doubt they are parenchymatous. But the characteristic feature of these cells is that they contain abundant amount of starch, that is reserve food. As large amount of starch or reserve food is present in these cells acts as a storage cells, and ultimately this zone is called as a storage zone or storage region that lies towards the ventral surface of this gametophytic thallus of Marchantia. Now, just below this storage zone is a lower epidermis, which is again a single layered thick, and large number of cells of lower epidermis produces a unicellular outgrowths like rhizoids, which are either smooth walled or tuberculated, or many celled or multicellular thin violet colored structures that are called as scales. So rhizoids and scales, they are the outgrowths that are produced from the lower epidermis. I have told you the function of rhizoids is like root, and scale that are present on the ventral surface usually performs the function of attachment, gives protection, and also it helps for the transport or conduction of water from soil to the thallus. Then next important point is a reproduction in Marchantia. Now this bryophytic liverwort Marchantia reproduces by both vegetative and sexual methods. Vegetative method takes place by two different ways, one that is fragmentation and second one is by gymme. This fragmentation is a very common when the thallus is growing in ideal conditions. That means when growth conditions are favorable for a longer time, this fragmentation usually observed in this gametophytic thallus of Marchantia. A process of this fragmentation is very simple. as the thallus grows, the progressive death of older parts, if you observe this, is a thallus growing here for a long time. The basal portion of this thallus usually dies, and due to the progressive death of this basal portion, the lateral branches or dichotomously formed branches get separate from each other, and each such branch again acts as a new gametophytic thallus of Marchantia. So this is a very common when this gametophytic thallus is growing under highly favorable conditions of the environment. Then second one is by Jimmy. So this production of Jimmy is the most common method of vegetative reproduction in a case of Marchantia. You cannot observe such characteristic reproduction, vegetative reproduction in other members of bryophyte. So it is a very most common method that is exhibited in this Marchantia gametophyte. Each such Jimmy is a lens-shaped structure. Singular Jimma, plural Jimmy are a lens-shaped structures, and they are produced in a large number in such a goblet-shaped or cup-like structure known as Jimmy cups. So these are the Jimma bodies or Jimmy, and such large number of Jimmy, they are produced in a cup-shaped structure. This cup is called as a Jimmy cup because as this cup produces Jimma bodies, so it is called as a Jimmy cups or Jimma cups. If you observe here, it is the section, vertical section of the thallus through the Jimma cup. This is as usual a thallus of Marchantia, which we have studied in internal structure. Only in addition to this, instead of a photosynthetic zone, here a cup-shaped structure is produced, and in the cavity of cup, large number of such a lens-shaped bodies are produced, which are called as a Jimma bodies. If you observe the structure of Jimma body, it has a short stalk and a multicellular structure. Here, this Jimma remains attached to the cavity of cup by means of a single cell called as a stalk cell, and from this stalk, this entire structure can get easily detached. If you observe the structure of Jimma, it has two such round structures. They are formed here due to presence of such a notch in the middle, and when it gets mature, this body gets detached from the cavity of cup, from the stalk, and when it falls on the suitable substratum, usually it began to develop into a new gametophytic thallus of Marchantia. Then second type of reproduction is a sexual reproduction. Here the sexual reproduction in a Marchantia takes place by fusion of male and female gamete, and this fusion of male and female gamete is of highly advanced and oogamous type. The Marchantia is a dioecious, or you can say it as a heterothallic. That means the male and female sex organs are produced on a separate thallus. Therefore, it is called as a heterothallic. Heteros means different, thallic means different thalli produces male and female sex organs, and therefore it is called as a dioecious or heterothallic. Here, The sex organs are usually borne on a special erect branches, each composed of a stalk and a terminal horizontal disc. Now, if you observe, this is a gametophytic thallus of Marchantia, producing such a long unbranched stalk, and at the tip of stalk you can have a disc-like structure. This disc is present at the tip of stalk, and whether it produces male sex organ or female sex organ, accordingly this stalk is called as anthridiophore and archegonium. Anthridiophore at its tip bears number of anthridia, and archegoniophore at its tip produces large number of archegonia, which is a female sex organ. If you observe the structure of anthridiophore, it has a lobed disc, and number of anthridia are usually embedded on the upper surface of each lobe. If you observe the distribution or production of anthridia, the oldest anthridium is found at the center, and towards periphery you can have the young anthridial structures that are produced on that disc. Here there are number of air chambers with air pores between the cavities containing anthridia, and at maturity the wall of the anthridium ruptures and number of biflagellate spermatozoids or anthrozoids are liberated in a drop of water. So here in this slide we can have a detailed structure of anthridiophore, archegoniophore, structure of anthridia and structure of archegonia. So here these are the anthridiophores having a long stalk and a disc which is having lobes. Anthridia, they are produced on the upper surface of this disc, and if you observe a vertical transverse section of this head of the anthridiophore, you can have a clear-cut internal structure like that of the gametophytic thallus, but in addition to this photosynthetic zone and storage zone, you can have a number of cavities that are present here, either in a photosynthetic zone or just embedded in a storage zone, and these are anthridial cavities. Now these anthridial cavities are producing anthridia, and each such anthridium is a small stalked structure. The stalk is a very short few-celled one, and the entire body of anthridium remains covered by means of a single layer or single layer jacket. And number of androcytes, they are producing the male gamete, that is anthrozoid or spore. Now here the mature anthridium or anthridial chamber is present at the center of this anthridiophore, and the younger anthridia, they are produced towards the periphery of this anthridial head or anthridial disc. Now when this anthridium matures, each this androcyte produces anthrozoid or spore. Each such anthrozoid or sperm is a spirally coiled structure, and this spirally coiled structure is having two anteriorly inserted smooth type of flagella, and when these mature anthrozoids, they are liberated outside this anthridial wall through this opening of this anthridial chamber, they lie in a water, which is essential for the process of fertilization, without which the act of fertilization in bryophyte cannot be observed, and therefore this group is commonly called as amphibians of plant kingdom. So these antherozoids, along with the water, it may be a dewdrop or it may be a rainwater, usually passes towards the female's sex organ, that is archegonia. Then this is a archegoniophore having such a long stalk, and the tip of this archegoniophore is not lobed like that of anthridiophore, but it is having a plate-like structure. So here you can observe the difference between the tip of anthridiophore and tip of archegoniophore. Here anthridia, they are produced on the upper surface, whereas archegonia, they are present on the lower surface at maturity. Initially they are also produced on the upper surface, but as this head of the archegoniophore grows, usually it bends down, and these mature archegonia, they are brought to the lower side of the archegoniophore. If you observe here, number of archegonia, they are produced in a series, and the mature archegonium, nearly mature archegonium, which is shown over here, is a small flask-shaped structure having distinct fainter and narrow neck. It is having cover cells at early stage, then neck canal cells, fainter canal cell, and a single large egg. But towards the maturity, here the lid cells degenerates or they get separate from each other. Neck canal cells degenerate, forming a passage for the entry of sperm, and there is a degeneration of ventral canal cell and presence of a single egg here in this ventral portion of the archegonia. Now, so these antherozoids or sperms, they are usually attracted towards this tip of archegonium, and one of the antherozoid enters into the neck of mature archegonium and ultimately reaches to the egg. The fusion of male nucleus and female nucleus takes place, that results in formation of zygote. And this product of zygote, which is a diploid one, divides, redivides, and produces next stage in a life cycle, that is called as a sporophyte. We have talked about this process of fertilization and so on, that results in the formation of a diploid zygote. Now, next point is a sporophyte. So, this sporophyte, this is the structure of sporophyte, is a structure developed from a diploid zygote after mitotic division. So this zygote undergoes number of cell divisions to form a sporophyte, and this mature sporophyte is clearly differentiated into fruit, seta, and capsule. Now, basal broad portion of the fruit forms form attachment. This is the basal broad foot that forms the firm attachment with the gametophytic thallus, where the venter of the archegonium was attached with this gametophytic portion, and this is only for to absorb the food material or nutrition from the gametophytic thallus, because this sporophyte is dependent on gametophyte as far as nutrition is concerned. So here this foot is a large bulbous structure that forms the firm attachment with the gametophytic thallus, and it derives its food from the gametophytic thallus. Seta is a elongated middle portion of this sporophyte, and it shows presence of large number of vertical rows having isodiametric cells. Then here this is an important part of the sporophyte, and it is called as capsule. Here this capsule is a round globular terminal part consisting of thread-like elaters and number of unicellular spores, and here these are the thread-like structures that are called as elaters, and these are the spore tetrads that are produced from a diploid cell after reduction, division or meiosis. So each such spore is a haploid structure. Each such spore is a haploid structure, and this entire sporophyte remains covered by means of a single layer jacket at the maturity. Now this sporophyte, entire sporophyte, remains surrounded by a layer of cells called as calyptra, and these elaters that are present here in this capsule usually helps for the dehiscence of the capsule as well as the dispersal of spores in case of Marchantia. Now here the mechanism of dehiscence of the capsule usually takes place due to the hygroscopic nature of the elaters that are present in the capsule itself. Here the sporophyte, which is totally dependent on gametophyte, and therefore it is called as a parasite on the gametophyte, and the spores that are produced here in this capsule towards the maturity liberates outside the capsule by forming a slit, and half of the spores, 50% spores, usually germinate to develop into male gametophyte, and 50% of the spores usually develop into the female gametophytic thallus of Marchantia. So this is about the sporophyte. Now each such spore after liberation are dispersed and carried away by the wind, and under suitable condition germinate by means of a divisions that are mitotic divisions. So large number of cells that are produced, and here 6 to 8 celled irregular filament first of all develop. Such 6 to 8 celled filament is called as protonema, and from a protonema a typical gametophytic thallus gradually develops or differentiates as a young thallus of Marchantia. So this is about the life history of Marchantia.